All right, now we're going to look at Chapter 2, which includes all of the medically important bacteria. We're going to start with just some introductory information for that, just looking at some of the properties of the cell wall and some of the different types of culture medium, etc., before we actually get into all the clinically relevant bacteria. Now, in this slide, what you're looking at are basically the difference between a gram-positive cell wall and a gram-negative cell wall. Clearly, the gram-negative cell wall is much more detailed. There's a lot more to look at there, and we'll talk about that. On the old school version of the USMLE Step 1, they would have shown you a picture of this and had A, B, C, and D, and E, and F pointing to different structures, asking you to identify that. But now that the exam is moving towards more clinically relevant exam, they're basically going to put everything in a vignette format for you. So I'll give you a few examples of how that might work, looking at cell wall structure. It's a stretch to ask a clinical vignette about bacterial cell wall, but we can do it. All right. So if you look at, again, the gram-positive structure, you can see where it's representative of the peptidoglycan, that on the gram-positives, it's very, very, very thick. Remember, that's what gives it its color in the gram stain. The thick peptidoglycan stains with the crystal violet and retains that color. Okay? Look over now to the gram-negative cell wall structure and notice that the peptidoglycan layer is very, very thin. All right? Again, pertaining to the gram stain, when you stain with crystal violet and then decolorize, that very thin layer of peptidoglycan isn't enough to retain that crystal violet. So basically all it does is counterstain with the pink or the safranin. Okay, so the peptidoglycan is very, very different. So that's probably a very important thing for you to remember about the differences between gram-positives and gram-negatives. Gram-negatives, in addition to having the inner membrane or cytoplasmic membrane, also have an outer membrane, all right? And it's the outer membrane that houses the endotoxin or the LPS. So the LPS for gram-negatives is actually part of the outer membrane. What is the toxic portion of LPS, you remember? Lipid A. Very good. Okay. Again, in the gram-negative, you have an inner membrane and outer membrane. In between, where you have peptidoglycan, is also called the periplasmic space. And this space basically has a sack of enzymes in it, helps regulate things into and out of the cell, something that you're not going to find in the gram-positive organism. Okay? So that's just a difference in the cell wall structure. How are they going to ask you that on the exam? They might say something like, you have a 5-year-old male presents with pharyngitis, fever, malaise. You do a throat culture and you find beta hemolytic cocci that are gram-positive. And they may want to know in the above infection how those organisms protect themselves against osmotic lysis. And in that case, they can kind of go back to the cell wall structure where you have to know that it's the peptidoglycan, in that case, a very thick layer of peptidoglycan that helps protect the cell. So again, it's a stretch. I'm with you. It's a stretch to go from a clinical vignette to the cell wall structure of a bacteria. But if I can do it that easily, they can do it too. All right. So those are some of the things that you might be looking for. Over the next few slides, we're looking at just some of the different components, just a little bit more specifically of the bacterial structure. Keeping in mind, again, the capsule is going to be on the most outer part of the bacteria, and 9 times out of 10, it's a polysaccharide capsule, which, remember, is sugary, is very slippery. The exception to that is the anthrax capsule that's made up actually of polypeptide. When you have something that's different like that, sometimes USMLE likes to pick on that because it's not like everything else, okay? So that can be a little bit of a trick, so that might be something that you keep in mind. The outer membrane, remember, is specific for gram-negatives, and this is where the LPS is going to reside. This is where you're going to have the porin structures, again, that allow transport of molecules into and out of the cell. We've already talked about LPS. The cell wall, etc., which is made up of peptidoglycan in gram-positives and gram-negatives, is just thicker in the gram-positive organism than in the gram-negative organism. So again, it protects it from osmotic damage. As we discussed, it's the basis for the Gram reaction. As far as, again, differences, one of the things you want to think of is you've got your gram-positives, you've got your gram-negatives, and there's one organism, here's your clinical relevance, that doesn't have a cell wall made of peptidoglycan. So you think about what you think that is, and I'll give you the answer in a minute because I know you're going to be sitting there stewing if I don't tell you. The reason I'm bringing that up is because one of the major targets for antibacterial drugs are the peptidoglycan and inhibiting the cell wall, the beta-lactam antibiotics, for example. In Mycoplasma pneumoniae, those organisms actually do not have peptidoglycan. So clearly, the easiest thing they're going to ask you, they're going to give you a nice short vignette about a kid with walking pneumonia, and they're going to ask you how to treat it. And you're going to want to put penicillin. You're going to want to put cephalosporin. But those target the peptidoglycan. And so you're actually treating the mycoplasma with erythromycin, actually, as a drug of choice. So that's, again, where some of that cell wall structure can come into play in a clinically relevant type of vignette. Now, periplasmic space, we mentioned when we were looking at the diagram, really kind of a sac of enzymes. And as you know, the inner membrane, the cytoplasmic membrane, is that phospholipid bilayer. It's very similar to what we would have in a eukaryotic cell. As far as surface structures, we did mention pili. We will be talking about sex pili when we do the genetics chapter, which is coming up next. So I don't want to get too much into that and get ahead of ourselves there. Flagella are surface structures of bacteria that give them motility. And whenever I think of motility, the number one bacteria that comes into my mind is Proteus. Okay? Proteus has so many flagella that when you try to culture it, it won't even really form colonies on the agar. It swarms across the entire plate. Okay? Now, axial filaments are specific for spirochetes. And the way you want to think about axial filaments, think about a spiral-shaped organism. So you got this little kind of corkscrew-like organism. The axial filaments help that corkscrew-like bacterium actually move and have that kind of corkscrew-like motion. So in a spirochete, and when we're talking spirochetes, really, for clinically relevant spirochetes, we're talking about Borrelia burgdorferi, which causes Lyme disease. We're talking about Treponema pallidum, which causes the CIF. And we're talking about Leptospira, okay, that causes leptospirosis. So in that case, motility is run by the axial filaments. All right. Internal structures, ribosomes, nucleoids, etc. You know the routine here. They're basically used in helping the cell replicate, make proteins, and very similar, and not exactly clearly, to what goes on inside of a eukaryotic cell, except for that there are no membrane-bound organelles. So that's an important point to remember.